Hello and welcome to the fifth episode of the NatSci Chronicles, the official podcast for the College of Natural Science and Michigan State University. My name is Jesse Early. I am the digital media manager for the college and the host for this podcast. The NatSci Chronicles is a podcast dedicated to keeping you up to date on all the latest happenings in the world of NatSci. In this episode, I'll be sitting down with two faculty members from the Department of Microbiology and Molecular Genetics to discuss their research with antibiotic-resistant bacteria. What are some of the challenges they face in their research and what are they hoping to learn and achieve? Stay tuned to find out. Today I'm joined by Victor DeRita, Rudolph Hugh Endowed Chair in Microbiopathogenesis, and Neil Hammer, Assistant Professor, both from the Department of Microbiology and Molecular Genetics within the College of Natural Science. Welcome both of you to the podcast.
Hey, Jesse. Thanks for having us.
You're welcome. Can you tell our audience a little bit about yourselves and your background?
Yeah, so I'll go ahead and start. I'm the oldest of six siblings. I grew up in the Chicagoland area. I attended a junior college prior to transferring to the University of Illinois. I earned a BS and MS at University of Illinois and worked as a laboratory technician in Chicago at Northwestern University in a tumor immunology lab. And it was about that time that I decided to pursue a PhD, encouraged by the mentor of the lab that I was working in. I earned a PhD from the University of Michigan. I hope you don't hold that against me. In 2009. From there, I trained as a postdoctoral fellow with Dr. Eric Scar at Vanderbilt University Medical Center. In Eric's lab, my research interest in Staphylococcus aureus began, and I was hired by the Molecular Biology and Molecular Genetics Department here at MSU in 2015.
Thanks, Jesse. I'm also from a large family, 5 kids, 2 sets of twins, so that's a little bit maybe
wow.
We got to be 5 kids. So, but I did my undergraduate work at Michigan State. I was in what was called in those days microbiology and public health, and then I did my PhD in plant biology, actually, and decided that I wanted to get back into questions of human disease and human health, and so I did postdoctoral work at Harvard with John Michelano studying pathogenesis of Vibrio cholera, which is still a threat throughout the world and was then and remains so now. And over the years, I studied a number of microbial pathogens. When I left Harvard, I went to the University of Michigan as well. I was there for 25 years in the Department of Microbiology and Immunology. It's actually where I first met Neil. And there I studied Vibrio and some other pathogens, including Campylobacter jejuni, which is a prominent foodborne pathogen, and Group A Streptococcus, which is a pneumonia and septicemia, skin, many manifestations of pathogenesis. And it was really only in the latter years before I joined the Michigan State faculty in 2015 that I started working on antimicrobial resistant, carbapenem resistant, in particular, Enterobacteriaceae, and we'll talk a little bit more about that. But I've been at Michigan State for about 5 years now and really enjoying it. I think the interdisciplinary nature of the work here and the combining, as we'll talk a little bit about, with plant biology and discovery for antimicrobials and just the way the structure of department work is really conducive to interdisciplinary research.
Great. Well, again, welcome both of you to the podcast. So both of you study and research antibiotic-resistant bacteria. Victor, one area you research is CRE, and Neil, one thing you focus on is MRSA. Can the two of you talk a little bit about each one, what that is, and what about them interested you enough to focus your research on them?
Yeah, absolutely. So MRSA, as some people might know, listening to the podcast out in the audience, MRSA stands for methicillin-resistant Staphylococcus aureus. And so that's the pathogen that I focus on in my lab. And two aspects of this particular pathogen that I find fascinating are intertwined. And that's the ability of the pathogen to colonize nearly every vertebrate organ and its remarkable metabolic versatility. So we and others have established that the transition between two entirely different metabolic states, one being aerobic respiration and the other being fermentation, promote colonization of host organs. And that's, in fact, pretty good news. So in terms of pathways that we need to inhibit to impede staph proliferation during infection, there are really only 2. And in fact, there's even better news. In the clinic, we are already pretty good at forcing staph into a fermentative state. And this occurs when we treat with a class of drugs called the aminoglycosides. However, the bad news is that a consequence of this treatment, therapies with aminoglycosides, is that it can result in these slow-growing fermenting Staphylococcus aureus that are resistant to aminoglycosides. And the resistant strains can lead to these persistent infections. So we call these things small colony variants because when you plate them on an agar plate, they're tiny. And I think that that's just really cool. So my lab focuses on identifying strategies to stop fermentative metabolism from occurring. And theoretically, these strategies will synergize with aminoglycosides in addition to help treating patients that are afflicted with persistent staphylococcal infections or those colonized with small colony variants. We should be able to potently, stop Staphylococcus aureus strains capable of performing both aerobic respiration and fermentation. So the idea is that we'll have whatever molecules that we discover that inhibit small colony variants treat in synergy with the aminoglycosides. And that's sort of the strategy that we're pursuing now.
Yeah, so we work with carbapenemase resistant Enterobacteriaceae, and that's a lot of words to mean it's essentially a multiply resistant microbe. Enterobacteriaceae is a broad class of pathogens, broad class of gram-negative, what we distinguish in bacterial genetics and bacterial physiology between two broad classes of microbes, gram-negatives and gram-positives. So MRSA that Neil just talked about is gram-positive. We study gram-negative pathogens, and gram-negative pathogens include things like Vibrio cholera and E. coli, many pathogenic strains of E. coli that we've all heard of, lives in our gut. And we're studying one called Enterobacter cloacae. And Enterobacter cloacae is also a member of the normal microbiota of the guts of a lot of people. But in some circumstances, it can become infectious, or it can become a human threat. And that threat usually occurs when people are immunocompromised or somehow other compromised in ICU. We see a lot of infections in the ICU of hospitals. And in fact, the group that I'm working with on CRE, and in addition to a number of other pathogens that are hospital acquired, are all looking at pathogens that were isolated from the University of Michigan hospitals. And so the broad group of pathogens we've commonly come to call the escape pathogens, and it's an acronym for a number of mostly gram-negative microbes that are threats to human health, primarily because A, they affect immunocompromised people very broadly, B, they're antibiotic resistant. And so what we're working on is pathways similar to the way Neil described MRSA, that are really metabolic pathways that contribute to the fitness of the microbe while it's growing within the host. So we're looking at different animal models to try to understand how does this microbe do its job of going from 1 to 2 to 4 to 8 and grow very aggressively within the host, just its physiological requirements, and can we intervene in those physiological requirements using new therapeutics?
Okay, so you guys may have answered this question a little bit here in your answers to the previous one, but looking at CRE and MRSA specifically, what are some of the challenges that they pose, and how is your research addressing those challenges?
Yeah, so this is a great question. It feels a little strange stating this during a global pandemic caused by a novel coronavirus, but in terms of mortality associated with antibiotic-resistant infections caused by all bacteria, MRSA has the dubious distinction of being the most lethal resistant pathogen in developed nations. So the rapidity with which Staph aureus becomes resistant is something that has interested me for quite some time. And of course, the challenge is stopping resistant organisms is something that we hope to achieve long term. And so working with Vic and others, we can not only stop MRSA from developing resistance to antimicrobials, but also potentially other dangerous pathogens as well.
Yeah, and so just to build off a little bit of what Neil said, one of the real challenges, and this sounds like a tautology or it sounds like I'm being precious, but I'm not. One of the real challenges with antimicrobial resistant pathogens is that all microbes eventually will, I say all, but for the most part, microbes will acquire resistance if we confront them with antimicrobial agents. Microbes divide a slow one divides every day. Humans divide every 20 years, perhaps, as we think about our generation time. So their generation times are what we struggle with. They can generate variants with population very quickly. And when we impose a selection, such as antimicrobial therapeutics, there's a lot of genetic space for the microbe to acquire and test resistant mechanisms and and those resistant pathogens will emerge out of that population as we apply therapeutics. So one of the things that we're working with here at Michigan State is developing compounds that are really not found in nature very broadly, so that these microbes haven't already been exposed to them over evolutionary time. One of the challenges we have with a lot of antimicrobial therapies is that for the most part, they come from other microbes. And so over time, and I mean evolutionary time, microbes have acquired mechanisms to become resistant to them. And all we have to do is put those roadblocks in front of new pathogens, and they will ultimately acquire it. So what we're doing with groups in the plant biology and pharmacox, maybe we'll talk a little bit about that, is to try to develop novel chemical space that these microbes may not have already been exposed to over evolutionary time.
Okay. So my next question here is sort of a two-parter. One, do you work with researchers from other disciplines? And two, if you do, how does it help to inform your research progress?
Yeah, so Vic and I are part of a strategic partnership grant. That means that MSU is invested in the research that my lab pursues, that Vic's lab pursues, but also the lab of several researchers on campus. And I think this is one of the strengths of MSU, is having such a breadth of research expertise across the campus. So in collaboration with folks like Edmund Ellsworth, Rick Newbig, Bjorn Hamburger, Jonathan Walton, before he passed away, we were developing novel therapeutics to test the very question of how can we find molecules that inhibit small colony variants in Staph aureus, and that can also be applied to some of to stopping CRE from also developing resistance. So just the collaborative environment of MSU allows us to pursue some of these questions.
Yeah, and it's really important to emphasize that, Jesse. So thanks for bringing that question up. We don't do science in a vacuum anymore. We're not Louis Pasteur in our basements with the ovens growing bacteria. We're all studying pathogens, excuse me, and antimicrobial resistance from a broad array of perspectives. And Neil pointed out the chemists and the pharmacologists and plant biologists that we're working with here on campus, and Rick and Edmund and Bjorn. We're also working, and I think Neil has done some work in this realm too, with biostatisticians and with bioinformatics people to survey genomes and tell us, here's some novel traits within a certain subset of pathogen genomes, and these might be things worth going after for drug development. So it's really incumbent on us, and this is, again, an emphasis of the quality of research at Michigan State, to reach out across campus and across disciplines to ask the questions that we want to ask. Everybody brings their own perspective to novel problems, and together those perspectives lead to new solutions.
Nice. So what do you believe is the biggest moment or finding in your research so far, and why do you feel that way?
Oh, this is a great follow-up question to what Vic was talking about in terms of collaboration. So I'm working on something, in addition to our work trying to find what makes the small colony variant fermenting Staphylococci tick. We stumbled across this really interesting phenotype in our collaboration with Dr. Sophia Lund in the biochemistry and molecular biology department. What it really allowed us to do was it opened up an entire research avenue in my laboratory that I never thought I'd be pursuing prior to coming to Michigan State. And so from a nutritional perspective, right, pathogens require various molecules to promote growth. And we've kind of touched on this in previous questions during this interview, but one nutritional element that has been vastly understudied, at least from my perspective, is sulfur, right? So this is a requirement of everybody. We need sulfur, plants need sulfur, bacteria need sulfur, and of course, pathogens need sulfur. But pathogens are limited to the host environment to obtain these nutrients. And so what my lab is trying to do is trying to understand how pathogens acquire this essential element during infection. And this is one of the more exciting avenues of research in my lab right now. And so we're using Staphylococcus aureus as a model to figure out how it acquires sulfur during infections, but we're also finding that some of these strategies are conserved in other bacterial pathogens, and we're pursuing those as well.
Yeah, it's an interesting question, Jesse. It relates for our work in CRE to some work that occurred during COVID. So I have a a very interesting student, Lucas Demi, who worked at Neogen before he came to Michigan State for grad school. He's always looking for new questions and new things to discover and new areas to go. And his work is on a class of proteins in a lot of pathogens that are interesting proteins that regulate a lot of virulence traits in a number of pathogens. And while we were sort of shut down over COVID time because we couldn't get in the lab in the early part of April and May, I couldn't get back until around June. Lucas did a really nice bioinformatic analysis of a certain class of proteins that he's looking at in Vibrio cholera to look at how broadly distributed they are and whether we could find new protein classes that might be regulating important metabolic traits for pathogens like CRE. And he found a whole group of these types of proteins in CRE. And so we're aggressively going after those. And this was all done by Lucas just sort of having downtime during COVID and just going into genomes and doing a lot of bioinformatics that didn't require him being in the laboratory. And he discovered a whole new class of proteins that we think are going to be interesting for us to go after. Now, those may never lead to anything related to antimicrobial resistant therapeutics, but it's new biology, and that's one of the other things that the reason why we're at universities is to uncover new knowledge. And so this is going to be something I think we're going to have a lot of fun with.
Fantastic. What kind of results do you hope to see over the next 3 to 5 years, and how can that be implemented to fight antibiotic-resistant bacteria?
Yeah, it's a great question. So identifying the mechanisms by which the inhibitors of Staph aureus small colony variants, how they inhibit proliferation. So I should mention that we've actually found a couple of molecules that are exquisitely specific to small colony variants. So they kill small colony variants while not killing staph that's aerobically responding. We think that that's really exciting. So trying to figure out how those chemicals are working, chemically refining or increasing their inhibitory activity, and of course, testing their efficacy in animal models of infections are all areas that we want to explore for sure. And then, to touch on the nutrient sulfur aspect, we want to continue increasing our understanding of the strategy Staph aureus and other pathogens employed to obtain this essential element. And then also observing how bacterial infection changes the abundance and spatial distributions of nutrient sulfur within the host is another goal of ours. So it's been demonstrated that for some nutrients, what the host does is triggers what's called nutritional immunity. So in terms of like iron, your body, in fact, contains a lot of iron. It just turns out that most of that iron is not available to bacterial pathogens. And we have some evidence that suggests that the host might be modifying sulfur in a similar manner where some sort of event, maybe inflammation, triggers response in the host to decrease the amount of sulfur within various organ systems. And that's something that we're very interested in following up on. So moving from the bacterial side to the host side. And of course, understanding the physiology of the pathogen while it's infecting the host is going to help us find molecules or targets and small molecule inhibitors of those targets to treat resistant pathogens.
Yeah, I think Neil pointed out something really interesting in our field. So, I'm a lot older than Neil, so I can say things like this. But 30 years ago, when we were studying bacterial pathogens, we were very interested in these really kind of cool, sexy mechanisms that they use to cause damage to the host, toxins and adhesion molecules that play a role in their ability to stick to some tissue, and secretion systems that look like syringes and they wiped out cells. And these are all really cool things to study as biologists. And certainly they expand our understanding of how pathogens work. But what we're really morphing into is, and going back in some ways, back to the future, is the original question that Francois Jacob said in 1965, the great molecular biologist, said that the dream of every bacterium is to become two bacteria. And that's true in the context of a host when it's causing disease. And what we're really learning about more is these interesting mechanisms that we studied 30 years ago are really important, toxins and pili and surface structures. But what's really important is the basic physiology of the microbe while it's growing within the host. So the great work that Neil's doing on sulfur acquisition, others of us are looking at other aspects of very fundamental questions of microbial physiology and how that impinges on the ability of the microbe to thrive in a host, and what the host does to try to stop that. This is a question of nutritional immunity that Neil brought up. And can we intervene in that conversation at a very fundamental level that is going on between the microbe and the host? So instead of going after these really interesting mechanisms that we're so excited about as biologists, let's just look at the fundamental physiology and target that. And I think the other thing that we're looking for in the next 3 to 5 years at Michigan State, Neil pointed out this SPG grant that the MSU Foundation funded with Rick Newbig and Bjorn Hamburger and late great Jonathan Walton, where what the plant people are doing is really creating new molecules that don't exist in nature, but are fundamentally based on what nature can provide to give us new tools to target some of these fundamental metabolic pathways. And I think in the next 3 to 5 years, through the work of Bjorn and Rick Newbig and Edmund Ellsworth, refining these molecules and giving microbial biologists like us and cancer biologists like Jamie Bernardin, who's looking at these same kinds of questions with cancer cells, can we defeat resistance to therapeutics? We're going to have some new tools and new compounds that are really MSU-specific compounds that the world, I think, is going to come and want to use, because these are really novel approaches, and we're pretty excited about the possibilities with these compounds.
So we've only touched on a small portion about your research here today, but is there anything else about your research that you'd like to let people know that we haven't already touched on?
I don't know, Jesse, these questions have been quite comprehensive. So I think you've pretty much covered all the bases here. I just want to say thanks to my research team out there, if they're listening, like really appreciative of all the hard work that they're putting in. Obviously obstacles associated with the pandemic. They've worked through, some trials and tribulations. We're back in the lab now, full strength. So we're ready to go. And I'd like to thank you too for having me on for sure.
Yeah, I think one aspect of all of our research that we have to emphasize here at Michigan State, and really at any academic institution, is the reason why we're at an academic institution, which is the opportunity not only to uncover new knowledge in a pretty unfettered way, as long as we can convince our peers on study sections to give us the money to do it, but also because we have the opportunity to train students and to train postdoctoral fellows. And so we could be at research institutes or at companies where the bottom line is the bottom line, and we're really trying to drive forward, and projects change very rapidly. We're at universities because we like the unfettered freedom of academic pursuit, but also because we have a chance to attract students and postdocs. And at Michigan State, we're very lucky to have great ones. And so I echo Neil in thanking my team. I mean, I spend most of my time at this seat in front of this window. This is not my lab, this is at my house. But my lab is doing 2 shifts, working their tails off in DPS. And so they're essential to everything we do. But that's true of all of us at the academic institution. It's our students and fellows that we're really excited to be part of their training, and that's a real blessing to be at an institution like this to do that. So thanks, Jesse, for your really insightful questions and the opportunity to talk with you guys this morning.
Yeah, and I've got just a couple more questions here for you guys. And speaking of students, if a student is watching or listening to this interview and is interested in contributing to the research that both of you are doing, what is the best way for them to do that?
Yeah, so they can feel free to send an email. My email address is hammern2@msu.edu. You can find that on the web. Google my name, MSU, it'll come right up. So yeah, feel free to check out our research. So the nutritional sulfur stuff isn't quite on the website yet. It might seem a little counterintuitive because I just stated this on a public podcast that's probably also going to be on YouTube, but we want to, not broadcast that quite as widely as we want to, but because it's still, it's still cooking. It's not quite ready yet. But so feel free to contact me via email. That's the best way to do so.
Yeah, and I'm at Dorita VI at MSU.edu. I have a son who's Dorita V1 at MSU.edu. So if you hit him up, he'll send it to me, but VI. And also I'm at Victor Dorita on Twitter, so you can get in touch with me by DM there as well. I also, if you're thinking about graduate school, then we have a terrific gateway program called the Biomolecular Sciences Gateway. And so I would encourage you to look at that, and this is how you join any of our labs who are doing this, not just Neil and me and Mike Krabowski. We have a number of really outstanding researchers who are asking similar questions in different pathogen systems, and also people doing great soil microecology, microbial ecology, and immunology. So there's an awful lot of opportunity for students, and both at the undergraduate and graduate level, to participate. And I will say, if you are a freshman or a sophomore and you're thinking about doing research, you should be actively reaching out to somebody to start, because it's a whole lot better for you, and you get a lot more done if you start early rather than coming in as a senior and saying, I want to spend the semester in your lab doing research. It's just harder to get you plugged in at that level. So start early and do it often.
So in closing, if anyone would like to learn more about you or your research, what is the best resource for them to go to?
Yeah, I would say the web. Just Google. You'll be able to find us for sure.
Yeah, exactly. Go to the MSU MMG site. We're also on Twitter at MMG_MSU. We're very active on Twitter, so check us out there. I'll also make a plug for any microbiologist who's watching us, is thinking to be a microbiologist, to look at the Society, American Society for Microbiology. And this is a shameless plug. Neil and I are both very active in the ASM, but this is a great way as an undergraduate to start creating a network of colleagues that you will be sharing research and experiences with for the next 40 years. And that's a scary thing for an 18-year-old to think about, but it's true. So look at the ASM as a great resource as well.
Okay, and with that, that'll wrap up our interview. I'd like to thank both of you for joining me on this episode of the podcast.
Excellent. Thank you, Jesse. Thanks for the opportunity.
Thanks, Neil.
Thank you. Once again, I'd like to thank Victor Derrida and Neil Hammer for joining me on this episode. You can find more information about each of them by heading on over to the Department of Microbiology and Molecular Genetics website at mmg.natsci.msu.edu. And that will wrap up the 5th episode of the NatSci Chronicles. If you'd like to support this podcast, please subscribe and rate us wherever you get your favorite podcasts. Be sure to also visit us on the web at natsci.msu.edu/podcast, where in addition to this podcast, you can also find our other podcast, the NatSci News Rewind. If you'd like to keep in touch with us on social, you can find us on Twitter at MSU_NatSci, on Facebook at MSU-CNS, on Instagram at MSU-NatSci, on YouTube at MSU Natural Science, and on LinkedIn, search for Michigan State University College of Natural Science. You can also stay up to date between podcast episodes with everything that's happening in NatSci by visiting our website at natsci.msu.edu. Thanks again for tuning in, and remember, making a difference, it's in our DNA.